Ever wondered how traits are passed from parents to offspring? It's a fascinating journey that involves a deep dive into the world of genetics. Today we're going to explore this journey, starting from the pioneering work of Gregor Mendel and going all the way to the extensions of Mendelian genetics. Gregor Mendel, a scientist in the 19th century, laid the foundation of classical genetics through his experiments with pea plants. He studied various traits like seed color, seed shape, flower color, and plant height. These experiments led him to propose two fundamental principles of inheritance. First, the law of segregation, which suggests that traits are determined by discrete factors or genes that occur in pairs. During the formation of gametes, these factors segregate randomly so that each gamete receives only one copy of each gene. Second, the law of independent assortment, which observes that different traits are inherited independently of each other. This means the inheritance of one trait does not influence the inheritance of another. Moving further into the world of genetics, we encounter the concepts of genotype and phenotype. The genotype refers to the genetic makeup of an organism, consisting of the alleles it inherits from its parents. On the other hand, the phenotype refers to the observable traits of an organism, which are influenced by its genotype and environmental factors. Alleles are also a key component of genetics. A dominant allele is expressed when present in either the homozygous or heterozygous condition. A recessive allele, however, is expressed only in the homozygous condition when two copies are present. To predict the possible genotypes and phenotypes of offspring from a genetic cross, scientists use a tool called Punnett squares. This tool is particularly useful in studying the types of crosses, such as the monohybrid cross, which involves crossing two individuals that are heterozygous for one trait, and the dihybrid cross, which involves crossing two individuals that are heterozygous for two different traits. In the realm of genetics, there's also specific terminology to understand. For instance, an individual is homozygous if they have two identical alleles for a particular gene, and heterozygous if they have two different alleles. Genetic homogeneity refers to when all offspring of a cross show the same phenotype as one of the parents, indicating homozygosity for the trait. A test cross is used to determine the genotype of an individual with a dominant phenotype but unknown genotype. As we move beyond the basics of Mendelian genetics, we encounter concepts like incomplete dominance, where the heterozygous phenotype is intermediate between the two homozygous phenotypes, and codominance, where both alleles contribute to the phenotype in a heterozygote, resulting in the expression of both traits simultaneously. There are also instances of multiple alleles, where genes have more than two alleles in a population, and polygenic inheritance, where a trait is controlled by multiple genes, resulting in a continuous range of phenotypes. In essence, Mendelian genetics provides a fundamental framework for understanding how traits are inherited and how genetic variation is maintained within populations. It's a fascinating world that continues to expand with new discoveries and insights, illuminating the intricate tapestry of life.